All this is Dr. Mobeen Sayed from DrBeen.com. Welcome to one more talk today. So the discussion today is going to be about a post which has two important topics in it. One is that there is a Nobel Laureate. What is his name? He's a French guy, so Montagnier. So he had done good work at one point, and that is that he discovered HIV virus. So there is a post going around, which is actually a fake post that says that Montagnier has claimed that people would die in 2 years. That is one, that is fake part of the post. Then there is another part which says that he has said that vaccination is a very bad idea. So that we'll talk about because he says it creates variants. So we'll talk about why vaccines do not create variants nowadays that we are seeing, and what can be the variants and how these can be produced. So we'll try to match up, can we produce a variant from a vaccine in a vaccinated person? And then there is another message that somehow antibody-dependent enhancement would occur, and that would cause infections as well. So we'll talk about these concepts. So of course, when we look at these topics, if they're not backed up by proper scientific papers, it becomes difficult. It becomes a rabbit hole to just go and figure out things. So I'll try to keep us as much sane and on the right track during this as possible. But there are some deep ends here. There are some not the best science that is here. So I'll have to kind of go through that then to be able to say this doesn't make sense. So please bear with me while we are doing this. So, and I think I'm going to continue to discuss these less than the best theories from various doctors or so-called experts in the coming days. So let's start. So this is DrBeen.com. There are a lot of links here. We'll start with Montagnier himself. This is his work. This is on the Nobel site, nobelprize.org. So Luc Montagnier, Nobel Prize in Physiology or Medicine, 2008. And he was born in 1932. The work that he did was very interesting. His work was that he found a retrovirus, which was later named HIV. So he found the HIV virus and its behavior. So retroviruses are viruses. Retro means the reverse. Are viruses whose genome consists of RNA and whose genes can be incorporated into host cells' DNA. In 1983, Luc Montagnier and Francois Bar-Sinoussi, if I'm pronouncing correctly, discovered a retrovirus in patients with swollen lymph glands that attack lymphocytes, a kind of blood cell that is very important to the body's immune system. So T cells, B cells, and other NK cells like things. The retrovirus, later named human immunodeficiency virus, proved to be the cause of immunodeficiency disease, AIDS. This discovery has been crucial in radically improving treatment methods for AIDS. So this is the contribution by Montagnier. Good contribution, good job. Now, here is where he stands nowadays. For example, a few days ago, or what is the date of this one? May 27, this is 2012. So not a few days ago. So in 2012, he said, DNA sequences that emit in certain conditions electromagnetic waves. So DNA sequences emit electromagnetic waves. And you know, when light is born or when there is light is electromagnetic waves as well that can travel in various kind of states. So it is possible that we have electromagnetic waves, but DNA sequences that are emitting electromagnetic wave and then saying the analysis by molecular biology techniques allows us to identify these electromagnetic waves as coming from bacterial species. This was never proved So this is a problem to try to disprove or discuss these things. We have to kind of take these on the face value and then go from there. So this was never proved. There was no paper, there was no publication, there was no peer review, there was nothing. This is just him saying it. Then this is a good article. I would love if you can go over this. The post that is nowadays circulating is a fake post. This is just like I had discussed a fake post that was named after Stanford's doctors, and that started this whole discussion. This is a fake post as well. However, he has done interviews lately which have gotten that vaccines make variants or there is antibody dependent enhancement. So there is some parts that are true and some that are fake. So I'm going to talk about those. So this is actually a decent article. Then here is snops.com, which actually does fact check with people. For example, if they feel that I have said something that is not correct and I'm important enough to look into what I'm saying, then they would go and do the fact checking and put the data together. So here they have done some fact checking on the messages given by Montagnier. So this is an interesting one over here. The message here was that somehow there are HIV. So this was a study. Let me show you the study first. Here is a study. The study was done, which was very quickly retracted. This was January 31st, 2020, last year's January, when the SARS-CoV-2 was in its beginning for the world. In here, so if you see here, Prashant, Pradhan, Ashutosh Kumar, and so on, these folks wrote this paper, and they said, we found 4 insertions. We found 4 insertions in the spike protein. Now we know spike protein very well, which are unique to 2019 CoV, SARS-CoV-2, and are not present in other coronaviruses. And then they say, importantly, amino acid residues in all the 4 inserts have identity or similarity to those in the HIV1, GP120, or HIV1 GAG. So these are the receptor binding areas or spikes, you can say. Then they went further and they said, interestingly, so you may have to now look at my hands for a second, and I'll show you the drawings as well. So they are now saying that, interestingly, these 4 amino acid inserts are at separate places on the spike protein, but when the spike protein folds, they all come together to work as a unit. So here, interestingly, despite the inserts being discontinuous on the primary amino acid sequence, 3D modeling of the 2019 nCoV suggests that they converge to constitute the receptor binding site, meaning SARS-CoV-2 had HIV receptors or receptor binding sites. This became an issue. Now, here is the interesting thing. They did this, then they retracted it. So look, this is February 2 that it was retracted. So if you see here, this paper has been withdrawn by its authors. They intend to revise it in response to comments received from the research community on their technical approach and their interpretation of the results. If you have any questions, tell us. So this was January, February of last year. But that seed that they left is still here, and it is growing, and it has become a tree and a plant, and we are just all circling around that there is HIV in the virus. This is very similar to other such seeds that were left and then retracted. For example, JAMA. So here we are talking about some folks, researchers, but think about it. American Medical Association, AMA, came out and said hydroxychloroquine should be denied when a doctor prescribes it. And they kept this stance for almost the whole year. They combined hands with pharmaceutical organizations as well, pharmacy organizations as well, and they combined their hands to say, if a doctor writes it, just refuse them, and the pharmacy association is and the pharmacists would be okay to do that. Then in November, EMA silently, in their own board meeting, acknowledged that they should not have said this, and a physician has a right for off-label usage, but they never made it public. So it is not just that here you're seeing something publicized and then retracted, and then the imprint or the foot marks or the thumbprint is still sticking in the society. There are bigger organizations like EMA doing similar things. You have one more such example, Surgisphere and WHO. Surgisphere came out with the hydroxychloroquine study that, hey, this is hydroxychloroquine is actually has no benefit. WHO, based on that, caused the hydroxy's trials to be stopped. And that fingerprint on the society in the whole world is still maintained while Surgisphere's study was removed. So this is a very similar thing. We still say that there is HIV in SARS-CoV-2. The original authors retracted that within a few days. So then there was another study which was peer-reviewed, which was shown to be accurate. And please remember, these folks have to either retract or leave it there, because doing sequences is not a rocket science anymore. So the folks who come up and say, well, SARS-CoV-2 has never been isolated, that's just another silly rumor from old times. Sequences and sequencing is very easy nowadays. So then it is easy to kind of verify when somebody is saying, I saw this. So here is another study which showed that there are actually inserts that those previous authors were talking about, but they were not really the inserts for HIV. And they were really not inserts. Insert means something added. For example, I take a book and add a page in it and then give it to you and say this was part of the book. These sequences of amino acids that they had seen were present there in the coronaviruses even before SARS-CoV-2, the other human coronaviruses, for years before. So they were not new sequences. Neither were they HIV sequences. They were similar. So it is just like this, that when you read multiple books, if I pick up these books, all of these books are going to... What happened, Lucy? What happened? You want to go to the other room? Give me 1 second. Come here. So Luffy actually wants me to go somewhere. So anyway, sorry, apologies. So I was talking about the inserts here. So they are looking at those inserts. So I was talking about the books. The words in the books can actually be repeating. You can find same words in many books. You can even find some sequences that are very similar. But you would not say that if you find a book, for example, Lange Immunology over here, and next to it is Kumar's Clinical Medicine, if they have similar sentences in there, you can't say that they are the same books. So here they looked at those 4 inserts, and they looked at other viruses, other pathogens, and SARS-CoV-2 and previous human coronaviruses and non-human coronaviruses, and they found that these inserts or similar inserts were present. I shouldn't call them inserts. RNA sequences were present everywhere, or genetic sequences were present almost everywhere. So here is their list of those. And then they said, second, these insertions are present not only in 2019 CoV. So again, calling them insertions is wrong because these are just sequences. Insertion is kind of devious, that something has been added. But also in 3 beta CoV sequences from beta 2 from 2018 and so on, and even 2013. So this was incorrect as well. That left its fingerprint on the society to the date. The correction never became so public as the incorrect thing has become public even when it was withdrawn. So truth is walking slower and falsehoods and lies are running around wildly. Then the idea of the strains. We will talk about all of these things in detail. I want to create a context in our mind here. This is the next strain, and I have been showing this from ever. that variants have been produced since the beginning. Look at this is December. This is 2019, and then here, original Wuhan, and this, check this one out. This is January 13, 2020. We have a new sequence from Nepal. Then this is January 23, 2020. We have a sequence from Hong Kong. And if you just continue to go on this chain, you would see that every day, every month, every time there are variants that are being seen. Then the idea of ADE, antibody dependent enhancement. Again, we'll talk about that today. But if you just wanted to see the whole discussion in detail, that what are the possible mechanisms of ADE, how many types of ADE is present, has that been observed with SARS-CoV-2 or not, you can actually watch this video as well. I have done that discussion in detail. I just saw a cool bean leaving a message here that he saw an ADE with SARS-CoV-2 in Cell. So I would look that up and update further if necessary. This is a good article about ADE, and I have discussed this article in this video. Then antibody-dependent enhancement in SARS-CoV-2. What is the chances? So we have done this discussion in the past as well. And if you see here, at the end of the day, we have not seen in actual human beings, we have not seen ADE occurring. Then this is another one. They have a wall, registration wall in front of them if the ad blockers are there. I have an ad blocker because my if the ads are present in the pages, they just cause so much churn that it becomes difficult to teach. So they have an ad blocker, so you have to be registered, but the link is present in the description. If you don't have an ad blocker, you can read it. And finally, there would be a touch for the Professor Dolores Cahill, who had said that we have become mutant and vaccines are going to cause those. So this is the holistic structure, context in which we'll talk about today. So get ready. Let's start. So this is the gift for fear-mongered humanity, the humanity who's kind of going nuts that, hey, what is happening? So Montagnier related message, brief history, Nobel Prize, we talked about it. Then 2012, he said there are what is that? There are bacteria that are producing electromagnetic waves. And sure, we can actually create genetic material that can emit electromagnetic wave. We can make genetic radioactive material as well for various testing in labs. That's not new. But bacteria just in general being able to do it is something that's also not correct. Then there was no publication, there was no proof. Even now when he says that there are variants, there is no proof. I think that Geert actually has a better way of presenting himself, is to say he has a sort of a nonsense paper, but at least he has a paper that you can go over that and say, okay, this doesn't make sense. Here, Montagnier has nothing. So his previous proofs didn't have anything. 2020, he says SARS-CoV-2 was built in a lab. Now, this one, I actually did not comment on this because I didn't see it built or I didn't see it not built. So how can I say it was built or not built? But this was connected to the HIV receptor theory that I showed you the article before, which was retracted. So people had used that paper to say those insertions in the spike protein of HIV sequences cannot just happen by themselves. These were deliberately and meticulously put there. And this is the Montagnier's point of view. He says the SARS-CoV-2 protein, the SARS-CoV-2 spike protein, is meticulously put together in a lab. So the sequences that are present in there, the theory around the sequence is debunked and wrong. Now, he says that because he saw that theory and he thinks that there are HIV pieces in there, there are no HIV pieces that have been proved so far. And was it made or not? I have not seen it, so I can't comment on this one. So he says he saw HIV sequences. I showed you. He said that it was meticulously added, so it could only be a human's work. And that actually is debunked as well. Again, we haven't seen it is done or not. So, for example, if HIV has a word, if imagine HIV genetic material is a book, there is a word in there. That word can appear in SARS-CoV-2 as well, although even that was debunked that it never appears. Then consider this, that even human coronaviruses are 86% similar to SARS-CoV-2, but it is still different. Then saying 3 or 4 sequences becoming from HIV would somehow make it drastically different, that is just weird. And then this is the discussion about that retracted paper, that they had said in this spike protein of the SARS-CoV-2, there were 4 inserts, which were then debunked that they're not inserts, they are part of the spike protein. They're simply genetic sequences that resemble somewhat, not even 100%, to other viruses as well, and bacteria as well, and cells as well, including some of the HIV sequences too. But they had made it shocking by saying that the receptor binding domain is made up of those sequences. That was incorrect. So they retracted that and they said, sorry, we agree, we should not have done it. But their message still percolates in society. Then I showed you the other paper that was peer reviewed, and that paper was accepted, and that had looked at the same sequences and said, hey, that's not the right thing, what the others are saying. Now the question, are vaccines, so there are 2 things left now in this discussion. Are the vaccines creating variants? And what is about the antibody dependent enhancement? So why not we go and address the antibody dependent enhancement first, because I drew it. For variants, I wanted to do more brainstorming together, so I left that out of drawing so I can more freely discuss it. So I'm going to leave this part for a second and go here to the antibody dependent enhancement. The basic idea of antibody dependent enhancement is the following. When we get infected with something, our body responds with innate arm immediately. Then it tries to amplify the adaptive arm of the immune system. Adaptive arm has 2 parts to it, humoral part and the cytotoxic part. Humoral part makes antibodies. Cytotoxic part kills cells that are infected. We have seen this throughout the year, this phenomena, this mechanism. That's why I did not draw it. The point with the antibody dependent enhancement is that if we are exposed to something and we make antibodies to that thing, in the future, if that thing arrives in us, let's use SARS-CoV-2 as an example, and as we make antibodies against SARS-CoV-2, and if those antibodies are not effective to kill SARS-CoV-2 or to neutralize SARS-CoV-2, then those non-neutralizing antibodies or binding antibodies will cause damage to us by allowing the pathogen to infect better and to cause destruction better. This is the basic idea of ADE. It has never been observed. So again, I said that one of the cool beans here said that in the cell.com, there is a study about ADE with SARS-CoV-2. So I'm going to go and check that out and update us later. But it has never been observed, ADE, to occur not only in humans, but in primates in general. ADE has been observed in specialized in vitro test containers where the environment is set up for ADE, or it has been observed by dengue and HIV in other animals and not in primates. Especially the SARS or the coronaviruses have not been observed in primates. And this study is done. I actually shared that study in the past and discussed it. Here is that article that actually shows and talks about those studies. And here is the video in which I explained it. So I don't need to explain it again, but I want to make sure that we understand. Now, how does that ADE play here? Let's say I got vaccinated or I got infected. So this idea of the people saying that, hey, the wild type infection is better than the vaccine, and they keep saying that vaccines are going to cause ADE, that actually is a nonsense idea. And the reason so let's try to give it a sense. The reason that they say it is sense, it makes sense, it is senseful idea is the following. They say, in the case of actual infection, our body will make antibodies against many parts of the virus. Right? This is why these folks like Cahills and Geertz of the world, they continue to say that, hey, let the infection rip through the society. Don't use vaccines. So this is what they're saying, that, hey, we have multiple antibody types. These would attack multiple parts of the cell, and that would allow a natural response and natural herd immunity to develop. On the other hand, if we just take the spike protein and then make antibodies against the spike protein and not to the rest of the cell, because the vaccine only makes the spike protein in the body, then when the actual virus comes in, there are two problems. One, there is the rest of the virus, which we do not know what to do about. And second, if there is a variation, if there is this is a condition, if there is a variation that defeats these antibodies from functioning and neutralizing, then we would now have, and I'm going to make an incorrect structure here. I'm just going to make an antibody hang here. So ideally, these are spike protein antibodies. They're supposed to hang with spike protein, but I'm just going to displace it just to say that now we have a binding antibody. It is not neutralizing it anymore. And that would allow the virus to continue to infect, number one. And number two, the binding part will become a problem because it is binding only and non-neutralizing. And I'll explain further. So the first problem in this discussion is the following. Assuming that the antibody that would connect or that we will produce, either infected or vaccinated. They don't go with infected because they're saying, don't get vaccinated, just get the natural infection. But the problem is natural infection is actually more dangerous. So I'll get to that in a second. So now they're saying that this antibody has to become defeated. The point is, when we are vaccinated or we had natural infection, we have hundreds of antibodies. And if one of them does become defeated, others are still working. That is one, and it's a very important point. This is why you're not seeing vaccines getting 100% defeated. And I'll go to the variants in a second, where we'll talk about the selection pressure and then the variant escaping the immunity. So I'm talking about the ADE for the time being. So here we're saying that, you know what, let's say this antibody is useless. The other antibodies are still functioning. So if they are functioning and they are neutralizing, then there is no problem. So then how do we reach a point where we can support the message by these folks to say, yes, you're right. So let's try to reach that point. To reach that point, what we'll have to say is the following. All antibodies that are produced by the vaccine, all, will have to become non-neutralizing. They will have to become converted into binding. How? Because the spike protein will develop wholesale change in it. So much change in it that all the antibodies produced by the vaccines are useless. How many antibodies? I think spike protein, it was about 7,000 amino acids long, if I recall correctly. And let's say 10 amino acid is one epitope, then 7,000 divided by 10. DrBeen knows that my math is wrong. So I did the wrong math this morning as well. So 700 antibodies. Let's say there are some overlaps as well, then it may be 1,000 antibodies. Let's say our body cannot make antibodies to everything. So let's say 100 antibodies, something. Make a number from this number of epitopes. Epitope is 10 to 30 amino acids long, and figure out what is the number of amino acids in spike protein, divide it with 10. That is, the antibodies are usually 7 to 10. That is the number of antibody types that will be produced against the SARS-CoV-2. And now we are saying that the spike protein will have to become so much changed that all of them will have to become useless. So that in itself is counterproductive, because if the spike protein has become so much changed, then it is not a spike protein anymore, because all of its functional units are gone. Then what is it going to do? This is like a something whose hand, let's say my hand works, and now you have just cut off the whole arm, and now you expect the little stem of the arm to work like a hand, and that's not going to happen. But let's say we still give this to say, no, what we'll do is we'll allow this spike protein to stay spike protein while we'll allow the antibodies to be defeated. So let's see how. We'll allow let's say the receptor binding domain, RBD, of the spike protein. It develops some changes in it that it hides itself from the antibodies. So antibodies cannot come in and attach with it. They can continue to attach with the rest of the spike protein of the coronavirus infected, the one that has infected us after the vaccine, but they cannot attach here at the receptor binding domain. And that allows the receptor binding domain to continue to work with ACE2, and now the neutralizing antibody is not neutralizing anymore. So the variant has defeated. And now what happens is that these antibodies have become the ADE proponents. What does that mean? What that means is we are saying that these antibodies that are attached to the virus now are going to actually become reasons for the virus to enter into our cells in a higher number. The antibodies become receptors for the cell. Those antibodies that are just sitting there, bound to the virus, but not doing anything. There are two problems in this one. There is a very simple problem that comes first to solve. So here is that problem. So let's say this is a whole new virus. This virus arrived in our body. Our body was vaccinated or was previously infected. Our body knew how to create the spike proteins. We quickly produce them. Those, I'm saying spike protein antibodies. We quickly produce the antibodies. Unfortunately, these antibodies were not of the right type anymore for the spike protein, because spike protein has changed. And we have given this clue, leverage here to the scientists who want this to happen, to say, fine, RBD still is changed but works. So virus is still able to work, while the antibodies are defeated but still bound. Right? So we have to do all this Pilates to make this thing work. So antibodies are still bound, but they don't work. Then we are saying that, hey, this is dangerous. But before we say it is dangerous, first thing to understand is that when an antibody is attached anywhere to a pathogen, antibody is a declaration of a threat and a marking of the pathogen as a threat, binding antibody. And it would start complement activation, and it would start phagocytosis. And result of phagocytosis and complement activation is destruction of the pathogen, not infection of the cells. So binding antibodies are actually very good antibodies as well. And ask any immunologist or virologist or whoever, say to them, do you think a binding antibody is actually useless when it is bound to a pathogen? Let's say it is not neutralizing it. That means it's not causing it from disrupting, from not binding. It's not disrupting it from binding. So it's not neutralizing. It's just bound. It's just bound. Is that dangerous? Is that bad? So first they will bring you here, and I'll go to that point as well, that, hey, this would cause ADE. But before we go to see why it would cause ADE, that is a theoretical mechanism, we should first see what is a biological function. And the biological function is when the antibodies are connected with a pathogen, they start the complement activation. They call the macrophages. Macrophages have a sweet tooth. That means they have a receptor to bind with the antibodies fraction constant and eat up the whole pathogen. And they don't just eat it up, and the pathogen is not just going to go and infect them. We eat up these pathogens in small pockets. We make phagocytes with them. Then we attach stomachs to the phagocyte and we kill the pathogen. If this was not the mechanism, we would never make it to this life, this age, 50, 60, 70. We would have been killed by the ADE type mechanisms in our young ages. So this is one problem to solve. So that means we have to give to such immunologists and virologists, we have to surrender one more normal mechanism, and that is a binding antibody will not activate complement and will not activate normal phagocytosis. So that means now this binding antibody is really useless. It doesn't do anything, and it is just sticking there, so that now we can cause problems with it. So you see how many things we have given up. Now, before I go to ADE, I want to talk about one more thing. The same folks who say don't take Moderna or Pfizer mRNA vaccines because this is what would happen, when you ask them that, fine, I will then take an inactivated virus vaccine because that has everything, that would not create useless antibodies. It would create useful antibodies to a lot of pieces. Then they say, well, that is really dangerous as well, because now we're going to create antibodies against M proteins and N proteins and other enzymes, and they are also just going to be binding. So the end result is what? They're saying don't get vaccinated, but get the infection. If you get the infection, not only there is a chance of death from the infection, there is also the same problem with the inactivated virus vaccine, which is with the infection. And that is there will be antibodies to many parts of the virus. So if that virus arrives again, you actually will have more useless antibodies than the antibodies against a spike protein. So we have to surrender this mechanism as well. To say, fine, we agree with you. That's also bad. So then we say, you tell us what is going to happen. And here is the theoretical mechanism of what is going to happen, which has never been observed outside of the lab systems or experiments or forced environments. So let's see that. First is this. So let's say this is a macrophage. Macrophage is part of the innate arm. Innate arm has macrophages, dendritic cells, B cells, natural killer cells, neutrophils, monocytes. Yes, right? All is good, Luffy. So these are the cells on the innate arm. I'm just going to take macrophage as an example. Here is a macrophage. The ADEs types, I'm going to go over all 4 or 5 types of ADEs as quickly as possible because I've done a detailed discussion before. One possibility is this. Look at this place here, this one. We have an antibody attached to the pathogen. Let's put the whole story together. I got vaccinated against the spike protein through a Moderna-like thing or Pfizer-like thing, mRNA vaccine. These folks are more especially upset about mRNA vaccine because they have a narrative there, and that is it's a new vaccine. And now they can make up lots of stories about it without, when they say, for example, when I got it, many people said, you are going to die. And I said, yes, I'll die eventually. But the thing is, if you're talking about future, you do not know the future and I do not know the future. So here, the antibody is produced. with the vaccine, and that antibody or antibodies are specific for spike protein. Then the actual infection occurs. When the actual infection occurred, the whole virus came in, which also has spike protein on it. Now somehow we now go back to the previous discussion, previous part of the discussion. We give everything, we surrender all mechanisms, and we just accept this part of the fact, or we make this as a fact to say there will be antibodies produced by our B cells. So here somewhere are sitting B cells. These B cells or plasma cells will produce antibodies. These antibodies will attach to the spike protein, but they will not be effectively able to neutralize it. This problem. They are attached, but they cannot neutralize it. Number one. Number two, they cannot do any other biological activity. And if they do any biological activity, it has to be negative. We have to surrender to all of this. So let's do that surrendering here. Antibody is attached to the spike protein. Antibody is not hurting the virus at all. Now you have a virus with an antibody hanging with it, spike protein, and the virus is just running around. Our body is not able to respond to this virus because the virus can still attach to the ACE2 and enter the cell because the antibodies are not neutralizing anymore. So virus is just running around, infecting the cells, coming out and doing stuff, while antibodies are just stuck with it, like a newly married couple's car that has those little strings on the back. Okay, now that antibody, so imagine this is one virus, this is the other virus. They are just stuck with this antibody. Antibody is not causing any harm. That antibody, the constant region is, so antibodies have two parts to them. There is a binding region with the antigen where the antibody binds with the offending agent. And then there is a constant region here, which is used for biological functions. Biological functions are what? Functions more than binding. If I ask you this, that, hey, your job is to figure out some dangerous, toxic thing and neutralize it, you're going to probably try to bind with it or figure out some way to capture it, and then some other way to neutralize it or kill it. Our body does that as well. Our antibodies capture the offending agent with one end. The other end is used to connect with cells and do biological functions. What are those biological functions? As I said before, most important biological function is to cause phagocytosis. So here is a pathogen. Here is an antibody attached to it. Here it is going to sit on a macrophage. Macrophage would then eat it up and phagocytose it and bring the pathogen in. And then in here in the vesicle, macrophage is going to destroy this pathogen, then present it to the immune system to work. That is a biological function. Or an antibody can activate complement system. These are proteins produced by liver, which are part of these proteins are part of innate arm, and these proteins can attach with an antibody. Then they can become activated, and they do two things. One, these proteins allow the macrophages, they're like receptor for macrophages, to eat them up, again phagocytose the pathogen with those proteins and then destroy the pathogen. Or this complement can start a complement cascade, which would make little pipe-like structures that would settle on the surface of the pathogen and make holes in it and kill it. These are biological functions of an antibody. None of these virologists, immunologists talk about the biological function, because if they talk about it, all of a sudden their whole palace or the house of cards falls apart. So let's go back here to our discussion. So now what happens is, this is an antibody. It has connected with the virus. And now, because it is not hurting the virus, at some point, this antibody is going to get connected to the macrophage or a dendritic cell. This is where the theoretical mechanism of ADE is possible. What is that? Macrophage is going to try to eat this thing, and the process is called phagocytosis. Phagocytosis is that macrophage, when it would sense that there is an antibody attached, it is going to invaginate that part of the cell membrane and try to make a capsule around that and make a phagocytic vacuole in which the pathogen plus the antibody is brought in. Then macrophage is going to combine this phagocyte with a lysosome, which is the stomach of the cell, which has acids in it, and then that the acids would destroy the pathogen. This is the normal mechanism. Here, what we are saying is, instead of this normal behavior of the phagocyte or macrophage, this thing will be internalized, but the virus will actually escape from that pocket, or the pocket will not form. And now the escaped virus has essentially infected the pathogen. This is like if a cop has put a criminal in the back seat, or a potential criminal in the back seat. Then that potential criminal breaks the windows or the car and runs away. So that is what has happened here. There are some pathogens that are known to do this. SARS-CoV-2 is not known to do this. We are offering it this possibility to say, hey, we are kind enough to you to think that you would do this too. So now this is one problem, that when we have attached to the pathogen, antibody is present, antibody has gone into the cell, and the pathogen has now become released. If it is released, now the cell is infected. And because the cell is infected, now the cell would go through the normal cycle of the virus replication, and now we have a problem. This is one mechanism. Second mechanism. Hey, Luffy. So the second mechanism is gene suppression. What does that mean? The other possibility is, again, this is a possibility. These are far-fetched ideas that we want them to be mainstream, but the actual phagocytic function, we want to surrender that to say that would not happen. The second possibility is that this antigen and antibody will be brought into the pathogen, into the macrophage. Inside the macrophage, the pathogen would run away by some mechanism. Then the pathogen would influence the nucleus of the cell and block the defenses. We know SARS-CoV-2, when it infects a cell, it does go through, it sends cargo through important alpha and beta into the nucleus and causes the nucleus to stop, reduce secreting interferons and tumor necrosis factors. So if this happens with SARS-CoV-2, then SARS-CoV-2, if it infects this cell, then it is possible that this would happen. The first given should be that we would say, yeah, this SARS-CoV-2 can enter macrophage through the antibody receptor, then become released inside and infect the cell, then send the genetic suppressors to the nucleus. So all that has to happen. That is another possibility of ADE. My request to you is that when you're hearing it, keep thinking about the normal mechanism was phagocytosis. And that is that antibody and pathogen are eaten up inside the cell, and then they are destroyed by the acid. That is the normal behavior of phagocytic cells. They're professional cells. This is their job. This is, for example, if we said to cops that we know your job is to arrest criminals or potential criminals. However, we believe that every time you'll catch them, you would release them. Or every time you'll catch them, they'll slip away from you. That is what we're saying to the immune system. We are saying, hey, man, every time you're going to catch this virus, it's going to slip away. And it's going to just infect you. It's going to kill you instead. So that's two mechanisms. A third mechanism for ADE is, look how far-fetched these mechanisms are. The third mechanism is that when the virus is connected with the antibody, it makes a change in the antibody, which is true, it happens. There is a concept of conformational changes in our biological systems. What that means is that sometimes, for example, let's say if you look at my hand, let's say this is an antibody, but it is not bound to anything. When you bind it with something, Then what happens is there are electromagnetic changes because there are amino acids that are positive and negative charges, and you have added one more set of amino acids. Imagine another set of magnets, and that is now going to change the shape of this thing. And this changing of the physical shape is called conformational change. So they're saying a third possibility is that a conformational change would occur because of the attachment with the antibody. That conformational change will allow, so check this out, will allow virus plus antibody to more efficiently enter the cell to infect it instead of becoming phagocytosed to be destroyed. So that is also a very far-fetched idea, but okay. When we have surrendered the basic concept of the right working of the immune system, then we have to kind of give all of this as well, that yeah, sure, now this all would happen. At the same time, another possibility of ADE is that as the antibody is attached on the surface of a phagocyte, and the antibody has a pathogen attached with it, that is, after the vaccination, B cells are present, actual infection occurs, B cell make the antibody connects with the spike protein of the new infection, the SARS-CoV-2, it's not able to neutralize it, then all other biological functions are muted, they're not being done, and now this complex, antigen-antibody complex, sits on the phagocyte, and instead of doing this, now we're talking about the complement system to be activated. And when that complement system is activated, the complement system itself also acts as a receptor. And it's actually a normal function. What is that function? The function is, our body does this very often. Let's say this is a pathogen. Our body would put an antibody on it, on its surface, or bind an antibody, binding antibody, which would then cause the complement to become activated. I keep making the complement here because on the antibodies in this area, there is a region where complement can dock and become activated. So complement becomes activated. This complement would settle on the surface of the pathogen like little candies. And when macrophage would come in, it would look at those candies and say, oh, yummy, I love candies, I'm going to eat this. And it has receptors for these C3, they are called C3b. It has receptor for these C3bs, and it is going to hold the pathogen with that complement and eat it up, phagocytose it, and then destroy it. So the next mechanism for ADE is that this process would also fail to phagocytose. Instead, this would also be used by the virus to infect the macrophage. Never been observed with coronaviruses, but this is another possibility. So these are all theoretical scares that this could happen. So that is the problem here. Complement acting as a receptor, but instead of helping with the phagocytosis, it is helping with the pathogen infecting the cell. Then there is one more mechanism related to complement, and that is that there is another part of the complement called C1q. When C1q becomes activated, then the C1q also acts as a receptor and a conformational changing system for the pathogen. So there are 5 mechanisms. Now realize this, there are so many folks who ask me daily, am I going to get ADE? Or people who are becoming sick, they are getting ADE. There are 2 important things to notice. ADE will not only, if it was occurring, it will not only occur with the spike protein vaccines, it would occur with non-spike protein vaccines, plus it would occur with the actual infection as well. You cannot tell the immune system. I'll give you, now let me transpose this example to infection. I got infected, let's say last January with the wild type coronavirus. I didn't, but I'm making up an example. Now we are here in the 5th month of this year, so 17 months later. There are new variants. My body's response to the original is not sufficient anymore. these have not become neutralizing anymore. Now I have binding. And my binding antibodies are going to be more than generated by the vaccine. Why? Because originally I had the whole virus. So my body created how many epitopes? 29,000 divided by 10 is the number of epitopes, compared to, let's say, 7,000 divided by 10. That is the number of epitopes. That means I'll have a lots of antibodies. Now we are saying that these antibodies have become useless. You tell me, after infection, I will have more useless antibodies if we follow this mechanism, or after spike protein, I'll have more useless antibodies. Generally, the basic answer is this. Our body does not just go mad and try to make antibody against every epitope. What our body does is that when a person becomes infected, we mount a response. So we make antibodies against 10 epitopes. And if that starts clearing the virus out, then our body does not bother creating more and more antibodies to more and more epitope. It says, fine, I'm clearing it out. I'm destroying it with these much. I'm just going to leave it like that. So with the actual virus, it's not that we'll have 29,000 divided by 10, 2,900 antibodies. We'll have a smaller number. But still, if we're going to talk about useless antibodies, they will be produced more with the actual infection, because actual infection is going to produce antibodies against all parts of the virus, which are going to be, majority of them are going to be useless. These are just going to be binding. So then the question for these folks is that, fine, you're saying that vaccine would do this. How about the actual infection? So then they take another position. And that position is, well, you know what? If you were not vaccinating, you will actually not have variants. And because you will not have variants, you would not have this problem. So then they need to be shown, and believe me, they know these things. It's not that they are that unaware. It's just that they know we can fool a larger chunk of people. And so that's what they're doing. So here, if you show them this and say, well, we started having variants from very early on. And believe me, that the variants that we have today are very far from the variants from the original. So why did we not make a noise at that time? Because in the beginning, media and folks did not know that variants can be something to be hyped up and create fear. And so in the beginning, variants were silently made. Now every variant becomes an issue. And there are variants that are becoming fast. And you know that the variant that would become fast would have to become less lethal. Or if it wants to be more lethal, then it will have to be slower. It cannot be both. But yes, I know that UK studies continue to say faster and lethal. And I'll explain what does that mean. So here are variants. Now, this is the antibody-dependent enhancement. You judge this. How much is this possible? Now, vaccines have been given for a long time. How many ADEs have you actually heard? It is because on daily basis I get comments. I know all the answers from the anti-vaccine side as well, that they would say, well, everybody who's dying is ADE. Or as Cahill said, she was very destructive, just like Geert is. Cahill said over here, everybody who dies, we should now start doing the autopsies on them to prove that they died because of the vaccine mutations. So then there are answers like that. Okay, back here. I hope this is clear that antibody-dependent enhancement is theoretically possible. Maybe it is possible, maybe in 1% or 2% as well, but it is not a mechanism that is the primary function of the immune system. Our primary function is opposite to the ADE. We use these antibodies to capture the virus, and even if we cannot neutralize it, we use it to capture it and do biological actions of phagocytosing it and destroying it. If this is an antibody which is IgA, in the mucous membranes, we capture it from allowing it to connect with the cell. So even when it cannot connect with the cell, so let's say it can connect with the cell because RBD is still open, because the antibody has become defeated. Even then, when the antibodies are coating a pathogen, it becomes very difficult for that pathogen to fuse with our cells and get into that. So the pathogen, it actually becomes very difficult. Imagine if we have, I'm going to draw this here. Imagine this is a pathogen and there are sticks, you know, connected with it everywhere. Even when those sticks are not able to neutralize it, it still has to figure out. So let's say this is the receptor protein, spike protein. It still has to figure out to go closer to an ACE enzyme to connect with it. And these sticks are just hindering. So IgA not only helps with blocking or neutralizing, it can actually just hinder the virus's capability to go near a cell and attach to it. So they don't talk about that. Okay, now I'm going to go to the final point for this one. This, in my opinion, saying that antibody-dependent enhancement is happening, we have been giving vaccines from what? Trials have been going on from August, September. And even before that, we've been vaccinating since November, December. People have been infecting from January. There is no difference in vaccine-related antibody and non-vaccine related antibody. Non-vaccine related or actual virus would be even more dangerous if we accept these mechanisms. But do you see antibody-dependent deaths? I have Cahill's transcript over here. She had said, and this is her previous year's talks, and she had said that next year, by February, March, April, there are bound to be new variants that are going to start killing people in mass. So we do have variants. Nobody is dying. Okay, so next, last point for today, and that is, are vaccines creating variants? The basic way to present this one is to look at what they're saying, what the folks are saying. So without talking about how our immune system handles it, let's first go to the idea of why they say variants will be produced. So here are the givens. The given is a person is vaccinated, one. Second, when the actual infection occurs, one. And we can give that. A person got vaccinated, I got vaccinated. I'm fully vaccinated, but still I cannot stop the virus to land in my mouth and nose. So I got infected. Third, my antibodies are not able to fight against it because it is a variant. So the problem is, if it is a variant, then it is already a variant. Then it's not my fault. I'm a vaccinated person and receiving a variant. So let's change that story to say antibodies are not able to fight against this because it is original type, but I'm making antibodies that won't work on it. So let's do that. So somehow we have to make this virus escape this vaccine first without doing a mutation. Because if it has mutated, then, for example, if it arrives in me and it is already mutated in the other person, and I'm vaccinated and it has escaped it, then that's a different game, and we'll talk about that. We're talking about the very first time creating a variant in a vaccinated person. So for that, first given has to be that the original virus arrives in that person. That person's immune system is not able to handle it. So the problem is, why am I not able to handle it? Because I'm vaccinated against it. So this is the basic fundamental flaw in the theory. But let's give this flaw. And we say, okay, fine. So here is original variant. It has a it has a spike protein that our body actually recognizes, and it would work. So let me give you a mechanism for giving this provision to the folks who make these theories. It would take our immune system 24 hours to wake up and attack this thing. These sleeping memory cells would take about 24 hours. So please, my apologies, I really need your attention this time, because it's a convoluted mechanism to reach a point to say now there is a variant, and it is an incorrect mechanism. So I have to kind of present an incorrect mechanism. So give me your attention. So here, original virus came in because the objective is to bring the original virus, and then because a person is vaccinated, create an anomaly in this person. Virus came in, immune system took 24 hours. During that 24 hours, we are assuming that the IgA here that is sitting in the mucous membranes, IgA is not functioning or is not present in the right quantity. How can that happen? So we have to keep producing these scenarios. This would happen when I got vaccinated today. 3 months later, I'm not infected again till 3 months. My immune system thinks, all good, this antigen is not coming in. I don't need to continue to produce more antibodies. Otherwise, I'll just flood this person's system with proteins of all kinds. So I should stop. So the immune system has created some memory cells in the shelf and then stopped making more antibodies. So antibodies have declined in the mucous membrane. There are not enough IgAs. The virus has arrived here. Ta-da, I'm here. Okay, you are here. We don't have enough IgAs. That allows the virus to infect a cell. So now virus goes from the immune system all the way inside the cell. Right? So you're looking at some givens here. One given, let's call this given to be a 90-day given. That this can only happen after 90 days. Not before, because the immune system continues to make antibodies for 90 days at least. In some people, 6 months, some people a year, then it goes to sleep. So let's say 90 days. Okay, so, and let me add one more color here. In UK study, they saw that after first dose of the vaccine efficacy continues to increase even after 84 days. So instead of actually going to sleep, it actually keeps building. But we are going to give this provision that this will happen, that after 90 days or X number of days, the person's my immune system is not making antibodies anymore. Cells are sleeping, new antibodies are not there. The coronavirus, original coronavirus, is able to get into my cells. Good. Now, when it has gotten into the cells, it has started replicating. So that is one mechanism that is occurring. Simultaneously, another mechanism that is occurring is that my innate arm cells, macrophages, dendritic cells, natural killer cells, neutrophils, that are sitting here as well, and that got exposed to the virus, either to the original virus that landed or the virus that is now coming out of the cells that it has infected. These macrophages, you cannot turn them off. This is why Geert did this thing, because he thought about this and said, okay, so people are going to say, like Mubins of the world are going to come in and say, you know what, macrophage would not let that happen. So he said, well, the macrophage is depleted. It's gone. The point is, so the flaw in Geert's discussion is, why would the macrophage go away? Why is it working for other infections but not for this one? Somehow macrophage has decided to close its eyes to this, or, as he said, innate arm is just depleted. If innate arm is just depleted, we would just die very soon for other reasons, other infections, not wait for the next SARS-CoV-2. But the simultaneous mechanism that is happening is that the innate arm is now picking up these viruses and phagocytosing them, destroying them. Remember, there is no antibody yet. So there is no ADE happening yet because the antibody generating cells are sleeping. So innate arm has picked up this virus, started to chop it up, bring it to the lymph nodes or other tissue B cells and T cells, the memory cells, and said to them that, hey, look what I got. And now they're waking up. This whole process is taking 24 to 48 hours. Meanwhile, the virus is propagating. Good. Virus is continuing. Now we have to give one more given in this situation. That one more given is the following. In these cells, the viruses that are now becoming prevalent, there are three kinds of viruses being made. And I've done these three kinds before, but I'm using this mechanism here to create a variant here. One is virus that looks like original virus. Good. So let's say original virus here, let's make this is original virus A. So the daughter cells are formed, daughter viruses are formed that are A, similar. Now, realize that this original virus is at risk of our antibodies response because it is original. We are trying to say original virus would go in a person and then variants will be created in a vaccinated person. So we have to bring in the original virus. So this is original. Find this original virus is at risk and all the daughters that look like this original virus are at risk. Fine. B, we will make one more virus that is really, really different. Its spike proteins are so different that they are kind of disabled. And this virus, B, is not going to go anywhere. It's going to die. Why? Because it just don't even know how to connect with the ACE2s anymore. So this is a doomed virus. Okay. Third, C, we make one more virus here that has some change in the spike protein. So this virus is one more virus in this one person, and this virus has some change in the spike protein. That some change is such, and it is so drastic that it will have to defeat the antibodies. This will become a variant, and let's see how much effort is needed for us to nurture it to become a variant. So let's say this is a virus. It has change in it. That change, how much change should this have so that this virus can escape this person? And the folks with the idea of selection pressure, the idea of selection pressure is that out of these viruses, a virus that would stay fit will have a chance to escape. That is selection pressure. Selection pressure is not this, that somehow because I'm vaccinated, then the virus inside me has some thought to say, I must produce new variants of type that can escape. It doesn't even know I'm going to produce antibodies. We are saying accidentally it is producing various kinds. And one kind is going to be the one that can escape these antibodies. Okay, so now let's see. If it is the original kind, our antibodies in 24 hours will be formed and they would destroy it. If it is a disabled kind, then that would not be able to work with anything and it would go away. Now we come to this area where we have a variant kind that is fit. Now for it to be fit and to be able to escape, how many things are needed? One, the changes in this variant have to be so much that now it would escape this body's immune response. So let's see what is the immune response that we want it to escape so that it can eject from the person's mouth and get out. Number one, we want this variant to escape the innate arm. Macrophages, dendritic cells, NK cells, neutrophils, monocytes. It has to escape them all. This is why Geert says, well, there is no innate arm, because if he accepts that there is an innate arm, then his whole theory falls apart. The problem is innate arm, we cannot live without innate arm. Even now, while we're talking, our innate arm is hard at work, picking up garbage, the cells are producing so much of garbage right now. There are RBCs being destroyed right now in our spleen. When I do this, RBCs are being destroyed. A macrophage that has eaten another 100 debris particles is dying. A neutrophil is dying. An NK cell is dying. They are living their normal life and they're dying. There are pathogens present everywhere, which is normal flora, and they need to keep in control. And the innate arm's function continuously is to clear up all of that. If you just remove the innate arm from me for one day, I will not make it to the next day. There will be so much of garbage collected in the body. There is so much RBCs collected in my spleen, which would need macrophages to clear them out, that my spleen would choke up. So back here, innate arm is sitting here, and it's going to attack it regardless of what is the mutation in it. It's going to attack all of them and try to clear them up. But of course, we give us this, that innate arm is not sufficient to be able to control the original infection or this infection, or the vaccination or this infection. Why? Well, if the innate arm was sufficient, then why do we need adaptive arm? Innate arm is never sufficient in most of the cases. I shouldn't say never. In most of the cases, it is not sufficient. It always works with the adaptive arm, and they both work together. So if you said, hey, this innate arm should be sufficient to clear out these viruses, that's not going to happen. That means it's going to try to clear out these, it's going to try to clear out these, and it's going to try to clear out these as well. So it's going to clear some of them. Fortunate for us that the innate arm will have training from a previous infection or previous vaccine, that it would have a better idea of how to work with this virus. But this virus, the mutated virus, is not cleared out fully. Fine. Now let's say this virus is now going to start ejecting from this person as well. So we are not at 24 hours yet. The person still has these viruses being generated, and they are ejecting from him as well. There are original ejecting from him as well. First important thing to keep in mind, this mechanism that is happening here will happen in a first-time infected person. It would happen more in the first-time infected person, because the first-time infected person would take 5 days to 17 days, about 9 days median, for them to actually become fully activated with the adaptive arm. Meanwhile, whatever is made will be shed. We cannot say that after the actual infection, the shedding will not occur, but after the vaccinated person, only the variants would just continue to shed. No, the original would shed as well for 24 hours. The variant would shed as well. And if the person was infected, originally they would shed. And if they were reinfected, their immune system is still going to take 24 hours to wake up. Immune system is not going to say that, hey, because I am a result of the original infection, I'm going to wake up faster. And this I can give you, that for every person, how fast the immune system wakes up is going to be different. Secondly, is the immune system already active or not is different. For example, somebody gets infected today, they recover 10 days later, their immune system is active for next 3 months. Within the 3 months they get infection, they would still have an active immune system, which would immediately within seconds respond. I have given a given over here, and that is we are beyond the time when immune system is active. We are saying immune system is sleeping. So that means immune system needs 24 hours, 48 hours, 10, 12 hours, various people, various numbers to wake up. So this person is originally infected by the actual infection or the vaccine. These things are going to come out, the variants and original. So don't you think that originally infected person will shed variant? Don't you think that a reinfected person with the original infection would shed variant and a vaccinated person in this time would shed variants? Yes. Don't you think they would shed original virus as well? Yes. Is this immune system's fault or vaccine's fault? No. The drama would start after 24 hours. Before 24 hours for a reinfection, all of the people are going to behave the same way. No fault of a vaccine here. Okay, so now let's reach beyond 24 hours. Or let me say X hours when the sleeping cells, T cells, B cells have woken up. And now they're going to work, right? So now they're awake. Here is the interesting part now. And keep in mind, vaccinated versus originally infected. I should add a third case as well, infected for the first time, but that would then take us in a different direction. So let's say originally infected and getting reinfected, or vaccinated and getting reinfected. The question to answer here is, are we going to have a variant produced? First thing where the disagreement is, if you go back to the folks who say that this would happen, variants will be produced by the vaccines, their first disagreement with me will be, no, you are wrong, innate arm will not work. Even if we give them this, that innate arm will not work, I would still show you that how this is still not possible. Innate arm not working in first 24 hours, variants will get out for infected and not infected. Question to answer is, will innate arm become disabled in vaccinated only? That will not become disabled. The Geared theorem says, if we can respect it that much, call it a theorem, says that when the antibodies start getting produced, then the innate arm cannot help because antibodies would do some things. That would take 24 hours. We're talking about innate arm now. So infected or not infected, innate arm will work. Now, the first disagreement, I'm trying to give every provision to these strategies, but first disagreement is innate arm will work. Okay, not sufficient to clear the virus. Otherwise, we don't need adaptive arm. Now, 24 hours have spent. The B cells and T cells wake up. And say, oh man, there is an antigen here that we recognize and we need to work against that. So, okay, this is the second part of Geard's theorem. And this is where this person, the French Nobel Laureate, he is saying that vaccines create variants. He did not give any paper. He promised a paper but never wrote one. So, and I feel sad that I'm talking about his nonsensical mechanism or non-existent mechanism. Let's say it is senseful, but we don't know what that is. It is in his head, instead of writing long haul protocol today. But I think this was important as well. So 24 hours have been spent. Now, after 24 hours or X hours, whenever that person's immune system, memory system has become awakened, the adaptive arm, here is what's going to happen. We would line up all those three once more. Original, disabled, and mutated, variant. Remember, the reason they're saying that the vaccines will give rise to variant is that they're saying vaccinated people, vaccinated individuals will not allow the original to go anywhere because we're going to kill it. So that would only allow the variants to survive. And this is why the selection pressure would allow the variants to be selected and they would get out. So we've already seen for 24 hours, everything will get out, not disabled ones, but I think you know what I'm talking about. Now comes the B cells time and T cells time. So let's first go to the B cell side. So here is a B cell, woke up, and he said, oh, I have seen this thing before. Now, it could be vaccine or actual infection, but let's go with vaccine because they are attacking the vaccine with this. So let's say here is the vaccine. Vaccine was spike protein based vaccine. Because of that, this cell only knows how to make antibodies against the spike proteins. And one cell only makes one type of antibody. So we'll have, let's say when the vaccine was given, there were 100 kind of antibodies that were generated. So that means there are going to be 100 cells that will wake up. One cell can only make one kind of binding antibody. So 100 cells will wake up. They would all start creating antibodies with their own binding regions. And they all have a different binding region. Now we have to give one more provision. So now what's going to happen is these antibodies that are being produced are going to start attacking all these three. Right? Normal behavior of the immune system. Now here is the time to create a variant. We are saying all these 100 people, immune cells that would make antibodies, they would be effective against the original because they know how to bind with the original. Their antibodies know how to attack the original. However, these variants, there must be a variant. Because this is a selection pressure, because there is accidental mutations happening, there are going to be, in this one person, there are going to be some variant, which is going to be so different that all 100 antibody types are going to fail on it. They all are going to fail. All cells will fail. Cytotoxic cells will fail. Helper cells will fail. Everything will fail. So here we will have to actually give this as a given to say, fine, we accept it as it is not possible, but we'll just accept it as a fact that, yeah, this would happen. So then what we are saying is, we are saying, Mr. French Laureate Nobel Prize winner person, or Geert, or some other people, we agree that the original virus will be killed because our immune system knows how to kill that. We also agree that this guy is not going to go anywhere because it is so changed that it cannot actually infect anyone. So even if it is so changed that our immune system would fail on it, but it is actually a useless virus. And then we're saying there are hundreds of more mutations created of viruses. And we see there is one mutation on which all of these have failed. All of them have failed. When that happens, then this variant, when that would get out, will be a variant that has escaped this person's immune system. Now, if that immune system was created by vaccine, then this variant has escaped a vaccine. If this was an original infection, then this variant has learned to escape the original infection. That is the basic gist of it. So now, before we give this given, that it is a given in their theories, before we give it, you tell me how many mutations have you observed in the variants? UK had 17 variants, mutations. Out of those, couple of mutations or 2 or 3 mutations were such that they would reduce the efficacy. P1 Manaus had some variation, which were again, I think 11 or 13, something like that. India originated variant 617 had 13, out of which 2 were more dangerous. Now there are 3 that are more dangerous. We are giving here a variant that would actually be a more changed variant compared to all the current variants that you saw in this whole year or 17 months. Plus, it will not be a disabled variant. It would still retain the functionality. And we are saying, I'm becoming the folks who are saying that the vaccines would create variants. We are saying that this mechanism will only appear because of a vaccine. This will not appear if a person was already infected. So let me extrapolate this for infected person. Infected person would have another set of cells as well. That would make antibodies on the other parts of this virus too, which these folks continue to say these are binding antibodies, these are useless, or they cause ADE. So now this is like having a cake and eating it as well, that on one end, they would love to say original infection would create more B cells that would have more antigens, and those antigens would attack this thing and kill it. On the other hand, they are saying that all the vaccine-generated antibodies are somehow unable to attack this and kill it. So let's talk about one more thing. Let's say this is a criminal. This is a criminal. The person is vaccinated. The B cells are producing antibodies, and somehow these are all antibodies not able to contain this from infecting more cells, creating more copies, and then creating enough load to get out. That means that antibodies that would attach with this thing's spike protein. So I'm making a bigger spike protein just for my attaching the antibodies. First, we are saying that antibodies cannot just bind at all. If that is the case, and this virus is not going to work. But let's say antibodies are able to attach, but they're not able to neutralize it. That means this virus will find ways to connect with ACE2 and still continue to infect while there are hundreds of antibodies hanging with it. You tell me, the biological function of an antibody, activation of the complement system, activation of a macrophage to eat up and phagocytose this pathogen, which we talked in the previous discussion. How would that all become neutralized? This is where, once again, Geert is sharper than the other folks by saying, well, there is no innate system. Because he knows not only innate system is going to cause issues here to his theory, innate system is going to cause one more issue here. And that is that once an antibody is produced, even if it is binding antibody, it is not neutralizing anymore. Even then, macrophages are going to come around and they're going to eat it up. They're going to connect here. They're going to eat this thing. And the binding antibody is going to activate a complement system, which is also going to attract macrophages to come and eat this thing. And binding antibodies are going to activate the complement system, which are going to cause complement cascade to become activated on the surface of the pathogen and kill the pathogen by creating holes in it. So now you tell me, how can this variant escape? And now let's create one more provision here. Created enough changes in it that spike protein is still useful but has defeated all the vaccine-generated antibodies. Then it has defeated the binding antibodies, so non-neutralizing, but binding antibodies biological roles as well. Let's say there is no innate arm. So there's nothing to pick it up. This virus is now just moving around in the body, having fun. Nobody's touching it. Eventually, this virus has to arrive to be shed in a respiratory system surface, and then it has to be shed from there by defeating IgA. So it has to first appear in the wet surface. So this is the wet surface. So this is a cell. This is a wet surface. So virus has to figure out that, hey man, I'm going to be here now, and I am just now going to wait for this person to talk, laugh, breathe, something. I'm going to become a droplet and get out. And while I'm waiting at my bus station to get out, I need my IgA to stay away from me as well. So we have to defeat IgA as well. So IgA developing B cells have to be defeated. IgG developing B cells have to be defeated. Innate arm has to be removed. Original virus has to be defeated, but the variant somehow has to escape all of this attack. Then, and this has to happen when the immune system was sleeping, so that it takes some time to wake up, during which time the virus becomes variants. Then, only then, this variant would get out. So I would leave it up to you to decide, can a variant do all those things? I'll give you context, and that is, variants that you are seeing today have been changing slowly over time in 17 months, and still only sufficiently changed to reduce the efficacy of some of the vaccines. And here we are saying a vaccinated person is going to create a variant that would disable every vaccine related antibody and still be functional and still be able to get out of their body. So I don't know, did I just so now the question is, I'm going to continue with the further discussion. Did I take 1 hour 38 minutes? Okay. I'm going to see if there is any question that I can attach here. Siddhartha says, amazingly thought and wonderful talk. Thank you so much. Is there any question left? Purple Lizard says, look at how busy Dr. Bean has been. I need a drink. I need a tea and vodka. Make some. Absolutely. Here's the problem. We have a bad spike. So let's see. Judy says, here's the problem. If we have a bad spike, no one will know why. They will say new variant, but is it causing worse infection because of exposure to previous infection, or vaccine? Is it worse variant? Look, Jody, a very simple thing is that you see it on daily basis, that folks say, well, the variants are produced with the vaccines. And if you would see, the amount of vaccination is not sufficient, but they still say it. So I don't think that here I can sit down and dismiss or discredit or answer every single message. And I have had with my friends these discussions. And I can tell you, for every point that is made, there is a counterpoint. But what is very interesting for me is lack of evidence. So lack of evidence comes from previous mechanisms from the textbook. I always ask my friends who challenge me on this one to say, let's open up a book, start from a book. Show me these mechanisms to be possible. Then let's go to research. Then show me those mechanisms to be possible. Then go to evidence. Show me the people who are vaccinated or originally infected to have been showing this mechanism. And it is funny that I continue to lump them together. This is the purpose of these folks, to say they are two separate kind of folks. But no, from an immune system's point of view, they're the same. One person is looking at a vaccine spike protein, and other person is looking at the virus. Now, I'll give you one more interesting example. If a virus, original infection, is going to be so bad, for example, calling long haul, by leaving spike proteins in a cell, then it is possible that if you're creating spike protein with the vaccine, that might be there as well. Now, it doesn't matter it is adenovirus-based vaccine or a Moderna mRNA-based vaccine or inactivated vaccine, because there are all kinds of objections. I wanted to answer this question: can a vaccinated person and I would tag on previously infected person, help build new variants because of the selection pressure? Answer is very simple. It's not going to happen. Or the chances of that is going to be the same as infected person and a person who is not yet vaccinated or infected. The chances are actually going to be more in them. Do you know why? I had deliberately left this part out, but think about it. I get infected for the first time. From day 1 of the infection to day 5 when I develop symptoms. Let's say we don't have variants and they're not speedy and they're not creating symptoms within a day. Here, I have a chance for the virus to do whatever. Do you think that the virus is going to be noticing that, hey man, person was not infected before, or this person didn't have a vaccine, so I should be kind and not make mutations? No, the mutations are going to form within everyone. And this person is going to shed mutated things more and for longer period of time because their adaptive arm is still not active and innate arm is not sufficient to clear it out. They would shed more possibilities of mutations compared to mutations being shed from vaccinated or originally infected, which are reinfected. So any questions? That TD says best lecture. Thank you very much. Time warp says we would be dead meat according to the Frenchman. Yes. So to be fair to the Frenchman, part of it is his message and part of it is people further twisting and adding on their own. So dying in 2 years, I was very, very much surprised. I said, how could he say dying in 2 years? Because there is no science that can prove that in 2 years somebody would die who got vaccinated today. I know that people say, you know what is going to happen, and people said to me, bye-bye, you're going to die soon, and so on. But 2 years, I was very surprised at how did he say 2 years? Cahill was sharper in this one because she said, as the new variants arrive, then the people who were being vaccinated in last year would die, which has not happened. So I was surprised about this part. And so when I looked at the messages, it turned out that part was tagged on by some sub-scientific mind who could not figure out that just the sentence makes it the whole thing wrong. So it was not him. But the remaining part, the variants creation I think ADE type things, especially the variant creation, is his problem. And he was wrong. Colin Hamill. Colin says, is it suffice it to say that variants will occur, but the less deadly will survive and those more deadly won't as they kill the host? So that is another thing. So the daughters that are occurring, similar to original, survive. If our body can kill them, or they would shed. Those that are too changed, they would not survive. They would not survive within my own body. If they're going to be shed to someone, they would go there and die as well. So no problem. Third type, those that can be more lethal or those that can be more transmissible. Transmissible more means in respiratory diseases, of course, they don't have wings to say, I'm going to fly more than the other ones. They're all the same. More transmissible are the ones that are less lethal, so they can continue to go without making a person too sick. So, Hemi, to answer your question, there could be two types that would be shed. One will be those that are more lethal. They would kill this person, they'll kill next person. And so they would very soon be gone. The viruses themselves too. And those folks who say that, hey, well, we have a 5 days window for them to silently go. No, that is wrong, because a virus that is more transmissible is going to cause infection faster. That is what you're seeing in the variants. So they don't have 2 days or 5 days. They now have 1 day. So their chance to transmit has become shrunk as well. So either they would disappear, those more lethal ones, or they will have to become milder to continue to survive. So sort of correct, Hemi. Morehouse says, "Oh my God, I'm going to die in 2 years. Got to run." So this Morehouse was somebody who just tagged on this sentence and named it after the French person. So Sweetheart says, where is the talk? So let me show you some links. This was a transcript for Cahill. I always get amazed when I look or read this one. This is the 2012. I have this link, the one that is opening up right now, fact check what apps. So this person, this is a fake post in his name. And then there are some interviews as well. This is an interview that are also published. And so those are present. You can see those links easily. In terms, if I had to rank the more damaging versus less damaging, or the more sharper versus less sharper, Geert was the most sharp one. He actually thought about that they're going to say what would happen here, what would happen here, and then he eliminated things. So for Geert, the questions that he will have to figure out how to answer are going to be, number one, that why would a normally infected person not do this while the only vaccine system would fail? Number two, why would the innate arm just be destroyed? Number three, when he says that only because the antibodies are binding only now, these have become leaky antibodies, then why would our immune system's remaining system not work? Imagine this. This is a virus arrived in my body, original virus. And because of vaccine, whatever is the magical mechanism, this has changed. And now my vaccine generated antibodies cannot figure it out. So it has become an untouchable virus in our system. You think that our system would not take it as a new thing and start attacking and preparing for it in a new way? Meaning our system, according to his theory, our system would just become hands up and say, "Me Lord, you are a variant and this person is vaccinated. We give you all the permissions to get out of this person and go do damage to others." That is the eventual thing. So there are so many things he has to explain. The point is, when people come to me and say, "You should go debate him," why do I have to debate a person who says 2 + 2 is 6? He needs to come up with references. So today, every single piece of talk that I did, I have references. Or you can come back to me and you can say, what books? And the books, Medical Microbiology, Javet's Microbiology, and the Lange. These are the three basic ones to start with. For ADE, I cannot show you the books because ADE is still a mechanism that is iffy. Some people say it happens, some people say it does not happen. It is still in the research areas. It has not gotten enough proofs behind it to become part of the books. Books can refer to this as, hey, there is something interesting here, but there is no such mechanism seen. HIV and dengue has seen it. So if ADE would get into textbooks, it would get into textbooks with HIV and dengue. Mbal is saying the best way to not spread any variant is to treat early. Correct. Okay. So then are topical nasal sprays like Star Pharma's viralize the solution to transmission? I don't know. I have to look at them. There are some topical nasal spray that can reduce the viral replication and bind it. So if the content is correct and if the mechanism is correct, they can help, definitely. Ni who says, the problem with the cytokine storm occurs in some people because their innate arm does not start fast enough. That's not also known. We just know that their adaptive arm, humoral arm, causes too much of the antibody production and activity. And then innate arm, macrophages are part of innate arm that just go mad. So it is actually a problem with the innate arm's over response by an incorrect cell type, and that is macrophage instead of a natural killer cell. So Jack says, what is up with the term anti-vaxxer? What business is it if someone takes a vaccine or not? That's completely up to them. It isn't anyone else's business. So I really, this is how I have been for the whole year, that it's not, neither is somebody going to listen to me if I said so, nor do I want to push anyone. This channel has been for a year just offering information or clarification of the concepts. That's all. What are the people's decision? That is up to them. Iskalka Quest says, did Griert say the innate arm would be destroyed? He did not say destroyed. As far as I heard from him, he said the adaptive immune system would become dominant in this case. Yeah, so tell me this. He said innate arm will become depleted. So let's go with the idea of adaptive arm will become dominant. What does that mean? Let's go back to our talk here. So here, I hope you followed this much. So what we are saying is for 24 hours, innate arm would still work, correct? Because innate arm has to work. Because the adaptive arm has not yet become active, so it is not dominant at all. So, and again, this is after the adaptive arm went to sleep. If you went back and said adaptive arm was active, so before 90 days or X number of days for some person, adaptive arm was active. Fine, adaptive arm was active. Did you bring in an original virus or a variant? To create a variant, you have to bring in an original virus because you want to blame this person for creating a variant. You cannot send a variant in him and say, oh, you created this, right? So you have to send the original virus. If you send the original virus in this person and his innate arm and adaptive arm were active at the same time, and they were active against the original, and he was vaccinated against the original, then he would take care of this one and destroy it. There will be no concept of creating a variant. So let's give it time. Let's say 24 hours later. So for 24 hours, innate arm was working, right? Because adaptive arm is not there to dominate it. So innate arm is working. Good. It is not sufficient. It is never sufficient. In some people, it is sufficient. In children, it is sufficient. People with better NK cells, it is sufficient. So there are cases. But in this case, it is not sufficient. So the infection is not controlled, and now original virus ended up and then created variants. And then adaptive arm woke up. You tell me this. You heard Geert with care. You tell me this. Did you hear some mechanism that would explain how all of the adaptive arm antibodies will become failed? And how innate arm will become less dominant? Let me now explain why that is a wrong concept as well. Thank you that there is somebody who has listened to him. So here is innate arm. Here is an adaptive arm. Good. Innate arm releases interleukin 12 most of the time. Adaptive arm in turn releases interferon gamma. This is especially T helper 1 pathway. T helper 2 pathway has other cytokines. Interferon gamma's function is to amplify the innate arm. Innate arm. So the question I'm going to ask is, please find me a mechanism that somehow the vaccinated person's adaptive arm can suppress the innate arm or become dominant over the innate arm. This is not a hand wrestling going on or arm wrestling going on that adaptive arm will win. What would happen is Innate arm would do its own function. Adaptive arm will become ready within 24 hours, proliferate, and try to create a lots of antibodies and try to clear it out. Innate arm will actually become amplified during that time as well, because when the pathogen is coated with antibodies plus coated with complement system. Have you ever heard from these folks talking about complement? Because it goes against their theories. The pathogen is now coated with antibodies and complement system. Innate arm will actually start eating them. This phenomena will be macrophage activation. So please do a simple Google search of macrophage activation, and you would find out that the macrophage activation is done by the adaptive arm. So where is the mechanism for the innate arm to become less dominant when adaptive arm is working? This is what is your homework. When the adaptive arm is working, these both arms amplify each other. Innate arm tells the adaptive arm to become amplified. Adaptive arm tells the innate arm to become amplified. Why? Very simple answer. Adaptive arm produces antibodies. Let's forget about cytotoxic for a second. And if you went cytotoxic, you said, well, Mobeen is trying to avoid cytotoxic because there would be something there. Cytotoxic arm is the one that actually produces interferons, which is an amplifier for innate arm. So if you went cytotoxic route, you would just lose. Let's go antibody route. When you go antibody route, antibodies that are now connecting with the pathogen, they're binding. I'm not saying they're neutralizing. They're not going to neutralize it. Fine. All 100 antibodies have failed for some reason. So now these antibodies are bound. They are doing two things. Number one, they are causing macrophages to eat up this thing. This eating up would cause macrophage to release cytokines, which would activate the innate arm. Not only activate the innate arm, it would go get more innate arm cells produced. Please look up GM-CSF production, granulocyte, monocyte, colony stimulating factor. This is just one of the factors. There are tons of factors that are produced. Go ask Geert that how would GM-CSF get stopped? Or ask him that why would an antibody that is an binding antibody, not neutralizing, somehow not be phagocytosed by a macrophage? Macrophage is saying, you know what? This person is vaccinated, and because Geert has sent me a message that I should not work in this person, so I'm not going to do phagocytosis. That's not going to happen. Other part, when the antibody is connected to the pathogen, not neutralizing, binding, it is going to activate complement system. Go please ask someone that why would the liver fail to make complement systems? And why would the antibodies fail to activate the complement system? So the complement system is activated, which would, number one, attack the pathogen itself by complement cascade. Number two, C3b would sit on the pathogen, which is also a phagocytic molecule for phagocytes, dendritic cells, macrophages, even B cells. What would stop macrophages now from eating up this pathogen? And when it would eat up the pathogen, it would release cytokines to amplify the innate arm. How would adaptive arm stop all this? Especially that adaptive arm that is created because of the vaccine antibodies. The given these folks have is the following. They circumvent all of this, not even circumvent, they don't even talk about it, that a binding antibody is still going to create a target for phagocytosis. A binding antibody is still going to create a target for complement activation, which has its own actions. And then this discussion is a secondary discussion to the first discussion of saying all antibodies in a vaccinated person will fail on these variants because somehow vaccinated people their antibodies would fail only, and infected person's antibodies will not fail. First, we have to agree on this. Then all of this is the discussion. So do you see how many flaws are in there? Okay. Let's move forward. All right, so I'm going to stop here. And we will continue tomorrow. I know that today's talk is going to make many people angry. So totally up to you to like it or not like it. But if you like, subscribe, and share, that helps me. And secondly, if you would like to support this work, there are 3 links in the description. You can buy me coffees, or you can support me through PayPal, or you can be a patron. So with this, thank you very much. And I would see you tomorrow. Tomorrow we'll have Dr. John Breen with us. Day after tomorrow, actually not on Friday, but on Thursday, we'll have Dr. Hector Carvalho from Argentina with us. Next week, we'll have Juan Cami, who has been looking at ivermectin use in India and has very good graphs. He's working with FLCCC as well. He's working with Dr. Tess Laurie as well. So he would join us and show us some data. Ivermectin has been doing great in India. So I think it is good to see that as well. So with this, thank you very much. And I would see you tomorrow.